Hello and welcome to this Royal Society Publishing video podcast. I'm Charlotte Wray and I'm here today at the Royal Botanic Gardens, Kew, with Dr. Martin Bidartondo of Imperial College in Kew Gardens and Professor Jeff Duckett from the Natural History Museum. Along with 4 other researchers, they've written a paper published in the Royal Society Journal of Biology Letters entitled The Dawn of Symbiosis Between Plants and Fungi. The paper discusses the primal relationship between mycorrhizal fungi and early terrestrial plants and reveals a previously unknown early symbiosis between a little-studied fungi subphylum and plants which were beginning to colonise the land 475 million years ago. Can you tell us a little bit about the current understanding of early plant terrestrialisation?
Right, terrestrialisation, about 480 million years ago. And we know that from different sources of evidence, from molecular evidence and from fossils. And we now know there were some really key features of plants when they got onto land. One was matratrophy, which is not a familiar word, but if I talk about placental nutrition, you'll know what I mean. All land plants have placental nutrition, just like humans do, which is surprising. What else? The appearance of conducting cells, in particular cells that conduct water to replace water that's lost by transpiration. Then they have cuticles, which are waterproofing. They have stomata, which regulate gaseous exchange. And probably the last of these really key innovations was symbiosis with fungi.
And how does your study alter the commonly held conception that the Glomeromycota achieved the first symbiosis with early land plants?
Well, I think it's probably about 30 to 40 years that the Glomeromycota have been regarded as the earliest symbioses. Different kinds of evidence. One is looking at fossils. The Wray chert in Scotland is an incredible sort of fossil deposit where plant remains were silicified, but very good fungi in these fossils, and these look like Glomeromycotes. Glomeromycotes are the most widespread in land plants, and as far as we knew before this study, they were also present in the liverworts, which are now firmly established as the earliest land plant lineage. Right at the bottom, the land plant tree.
Why do you think that previous studies have failed to identify the relationship between Mucoromycota and early land plants?
I think there are 2 reasons. One is that they didn't use the right molecular techniques, and Martin explained that, and the other, they looked at the wrong organisms. This is a liverwort, Conocephalum conicum, collected from the gardens here. It's a weedy species. The sequence from the fungus from this, which is in the paper, is actually a Glomeromycote. So all the fungi from liverworts that have been sequenced previously have been from liverworts which are easy to get, like one's growing at Kew and one's growing in greenhouses. In order to look at the really interesting liverworts, you have to travel a bit.
Could you tell us a little bit about the plant samples that you used?
Right, these are liverworts with a predominantly southern hemisphere Gondwana land distribution. We knew from previous cytological work I'd done with Roberto Ligrone, who's one of the authors of the paper, that the cytology of the associations in these early liverworts, Tribia and Haplomytrium, are different from typical Glomeromycotes. In the typical Glomeromycotes, the fungi are always intracellular. Well, in these 2, they're both inter and intracellular, so they're rather different, and structurally they're rather different. So we were keen to see what we might find. What we were hoping for was that there might be different Glomeromycotes from those which had been described previously. Of course, what we found was completely different and unexpected.
And Martin, can you explain to us what mycorrhizal fungi are?
Mycorrhiza means fungus root, and it's unfortunate in a way that it is such a difficult word to pronounce and to write even, because it is actually very common. Almost any plant, 80–90% of plants all over the world form mycorrhizas, and in fact some people say that you shouldn't say that plants have roots, you should say that they have mycorrhizas, because it is very common to have fungi that are growing inside of the cells of that plant or in between the cells or both, and then are going out into the soil. And the fungi are very well adapted at taking up mineral nutrients and water from the soil. Plants can do this as well, but the fungi are often better because their filaments are thinner and they're longer than plant roots or plant root hairs. So there is an obvious sort of amount of soil that is greater that you can explore when you're a plant that has access to this fungi versus a plant that doesn't. For many of these fungi, the association has become obligate, so that they require association with the plant to complete their own life cycle.
What methods and data did you use to construct the phylogenetic tree shown in the paper?
We started looking at these plants and they very quickly fell into two groups. There was one group in which I could confirm that they were associated with Glomeromycota fungi, as expected. And there was another group that was the group that we were interested in because they looked different, and I couldn't find anything. They just didn't seem to have anything that I could detect. So I had to sort of stop, think about it, and backtrack a little bit, and sort of not make so many assumptions as I was making, and started to just say, okay, well, they are fungi. Let's just see who's there, and that is difficult because it becomes very laborious because there are always very many fungi in environmental samples. So then I was able to eventually figure out, well, they are a different group of fungi. They are not glomeromycetes, and that was done by obtaining sequences from the fungi and comparing them to each other. That created the second problem. It's like, okay, well, are they that group of fungi or are they not? So then I contacted a colleague that is an expert on this group of fungi. He's probably the only one, Jim Trappy from Oregon, and he provided me with some reproductive structures of this group of fungi, endogony. In that, I was then able to obtain sequences from them and then compare them to the sequences that were coming from the plants. And so then that allows you to create a phylogenetic tree where you can have both sequences from named specimens and sequences that come from environmental samples that you don't really know what they are. And if they are sort of interdigitated in a way within the tree, then you can sort of make the inference that that is the group of fungi that you have.
And how do you think that your findings will affect future work in the field?
Oh, it means a lot more foreign travel, I'm afraid. We could have to go back to the drawing board, and you can't, for this kind of work, you can't just rely on things that you can obtain from the nearest greenhouse. You really have to go back and search in the wild. Southern Africa, northern India, Mexico, where nobody's ever looked. So and we also need to look at some of the more of the ferns and lycopods too, because it's likely that some odd fungi are going to turn up in these as well.
We have been focusing for this analysis on what so-called lower plants, but we have found some of these fungi in ferns, and we have there is evidence that some of these fungi are able to associate with higher plants as well, in a different form of mycorrhizal association. So perhaps we need to start looking at higher plants as well, within those locations and in other locations.
And what's your next step in examining the early relationship between fungi and plants?
Basically, it's going back to the drawing board and doing a lot more samples. And then we've teamed up with Sheffield University. Now, Sheffield University have the most wonderful growth cabinets in the world. David Beerling, these wonderful cabinets, in that you can grow plants under different atmospheres. Now, when these plants evolved, carbon dioxide levels were about 4 times what they are now, about 1,500 parts per million. And what we're going to do is to grow all these primitive liverworts in these Paleozoic atmospheres with and without the fungus. Now, the hypothesis is that because you've got high CO2, it should drive symbiosis, and it's more efficient to photosynthesize when you've got more CO2, so they can afford to give more sugars to the fungi. So we're going to try and simulate these conditions, which is really, nobody's ever tried to do this before, so it's really remarkably exciting.
Thank you, Martin, and thank you, Jeff. And thank you for watching this Royal Society video podcast.